Hello everybody, let us discuss about the module 2. In module 2, first we will discuss about the marker assisted breeding. In the last lecture, we have talked about the conventional plant breeding method, but nowadays most of the breeders follow the marker assisted breeding. So, before going to the topic, first we have to know what is a marker. It is an indicator to identify or highlight anything. It may be anything. Suppose in a particular paper, a lot of sentences are available, and using the marker we have highlighted certain parts. So, it will be easily visible. Nowadays, those markers are highly used in plant breeding, and those are of three types mostly. First one is the morphological marker. Morphological markers are those which we can distinguish by observing it, like the flower color, like the plant height. Okay. These things can be morphologically distinguished. Suppose we have different rice varieties, some of them are taller in nature, some of them are shorter in nature, dwarf in nature. In this way we can distinguish it. Here you can see the flower color, two different flower color of Lathyrus species. Okay, one is blue, another one is white in color. So, it can be distinguished. Then along with morphological marker, other markers are there like biochemical marker. Suppose we have 4 to 5 different wheat genotypes. By seed we cannot distinguish it easily, but if we use the biochemical markers at protein level, at isozymes level, we can distinguish them. So, different bending patterns can distinguish one variety with another one. And last one, which is highly used nowadays, is molecular marker. And in molecular marker mostly we deal with the variation at DNA level. Okay. So, these are some examples of normally used molecular markers in plant breeding. First one is the RFLP, that is restriction fragment length polymorphism. It is a hybridization based molecular marker, restriction digestion and hybridization based molecular marker, I will discuss later on. Then RAPD, random amplified polymorphic DNA. Then AFLP, amplified fragment length polymorphism. Then SSR, simple sequence repeats. It is a very popular molecular marker and it is a PCR based technique, PCR based marker. Then ISSR, inter simple sequence repeat. Then CAPS marker, cleaved amplified polymorphic sequences. SCAR marker, sequence characterized amplified region, and SNP, the single nucleotide polymorphism marker. So, let us discuss about the RFLP and Southern blotting techniques. Okay. So, RFLP, as I mentioned, that it is a restriction digestion and hybridization based technique. So, in RFLP, let us assume we have two different rice plant, one is P1, another one is P2. In rice plant, suppose a particular chromosome has been shown over there. In that chromosome, in case of P1, 3 EcoR1 restriction sites are available. Now, what is restriction sites? Restriction endonuclease are group of enzyme that can cleave the DNA at a specific sequences. So, if those enzymes are used, the DNA will be chopped at that position. So, in case of P1 plant, at 3 positions the DNA will be chopped, whereas in case of plant 2, only at 2 positions the DNA can be chopped. But in the third place, means where a star like things is available, there some random mutation have been taken place, and due to that mutation the DNA sequence have been changed, and if the DNA sequence is changed, the restriction enzyme cannot cleave the DNA. So, in this way, suppose naturally we have two different rice varieties, in one of them after restriction digestion we will get 2 fragments. In P1 we will get 1.5 kb fragment and 0.5 kb fragment, whereas in plant 2 we will get only single fragment 2 kb. Okay. Now, if we do electrophoresis by using this digested DNA product, then after restriction digestion with EcoR1, in the left side we can see that in plant 1 two different bands are available, whereas in plant 2 only single band is available. Now, we have to go for southern hybridization. So, doing hybridization, what do we need? We need a particular probe. Probe is nothing but it is a known sequence which will be labeled first either with radioactive molecule or with non radioactive molecule, and thereafter that labeled DNA sequence will help us to identify its presence or absence, okay, or its presence at a particular position. So, in the right side where different hybridization pattern has been shown, in case of P1 we can see only one band like 1.5 kb, because here the DNA has been cleaved due to presence of the intermediate EcoR1 site in plant 1, whereas in plant 2 no digestion was there inside. So, we will get a band at around 2 kb. So, in this way we can see a polymorphism between different genotypes. So, in this way the southern hybridization and RFLP basically works. Now, let us discuss about the SSR or simple sequence repeats and its application. From the name itself we can know that these are simple sequences. It has other name also like short tandem repeats, the microsatellites, etc. So, it may be of different types. In our genome different SSR or microsatellites are available. Some repeats are mono repeats like a single basis A or G 5G, 5A, 6A are available. Okay, those are known as mono repeats, means single base is repeated several times. Sometimes there may be di repeats, means only two bases A and T are repeated 3, 4, 5, 10 times. Some tri repeats are available, like here ATC is available for 4 times. In this way tetra repeats, hexa repeats, different types of repeats are available. Now, how this sequence difference could be utilized in plant breeding? Let us discuss about that. Suppose we have 4 different varieties, one is V1, another one is V2, then V3 and V4, 4 different varieties are available. In variety 1 the ATG is available 6 times, in variety 2 the ATG is available 5 times, in variety 3 it is available for 4 times, whereas in variety 4 2 ATG sequences are available in tandem manner, means one by one, okay? No gap is available in between. So, definitely these 4 varieties are different, but normally we cannot distinguish it. At molecular level how we can distinguish it? Let us see that. Through PCR or polymerase chain reaction we can design different primers and we can amplify the region in between these 2 primers. As we have shown here in case of variety 1, 2, 3, 4, different forward primers have been designed from the blue region and the reverse primer have been designed from the green arrow region. So, if we use those primers, forward and reverse primer, for each variety separately, then we will get different sized amplicon, means primer will amplify that region, the in between region, and in different variety we will get different sized amplicon, like in case of variety 1, 2, 3 and 4 different amplicons are available. So, those were known varieties. Now, if we get some unknown genotypes of that particular plant species, in those genotypes also we can use the particular set of primers, SSR primers. So, there we can get the PCR amplification and we can see some banding pattern, and based on that we can say that the unknown genotype X2 is similar to V4, the unknown genotype X3 is similar to V2, and the unknown genotype X1 is similar to V2. In this way we can distinguish different varieties based on their amplification using SSR markers. Now, coming to the applications of molecular markers. First of all, it is used in phylogenetic study. Let us assume we have a hundreds of rice germplasm, and we need to know that which genotypes have been derived from where. So, this analysis, this phylogenetic study can be done by SSR and different molecular markers. Then the trait identification and mapping of QTL or quantitative traits loci can be done using molecular markers. Okay, that which marker is associated with which particular trait can be identified. Then gene mapping can be done, the marker assisted selection can be done through molecular markers, what we will discuss later on. Then DNA fingerprinting and molecular diagnostics can be done, and seed testing of different varieties can also be done using molecular markers. Let us assume that two plants P1 and P2 have been crossed to make a single cross product, whereas P3 and P4, these two plants have been crossed to get another single cross. Now, we are developing different double cross by crossing the left side single cross and the right side single cross. So, in our offspring we can trace a particular gene or a particular sequence that from which plant P1, P2, P3 or P4 it has come, if we know the information of molecular marker. So, in this way the markers can be used to trace a region from parent to offspring, how a particular sequence is transferred from parent to offspring. Now, let us discuss about the application of molecular marker. First of all, in our last class we have discussed about the conventional backcross method. What we have done in backcross method, we had a particular variety which is superior in nature, it has good agronomic characters, it is good yielder, but it has certain lackings. So, that lackings was improved by backcrossing with another variety which had some resistant trait. Here also we will show the marker assisted backcrossing method. So, it will be comparable, that how the molecular marker will be too much useful compared to the conventional plant breeding method. So, here the P1 variety is a drought sensitive high yielding cultivar, like which was used as a recurrent parent over there in the backcross method. So, it is a drought sensitive high yielding variety, whereas P2 is a drought tolerant cultivar, but it is a poor yielder, but it has an interesting feature that it is drought tolerant in nature. And by molecular study we came to know that the red highlighted region, that sequence is necessary for the drought tolerance, and we would like to transfer that specific region into this plant's genome, into the drought sensitive plant's genome. Thus, the red part should be transferred because it is a molecular marker associated with drought stress, drought tolerance. So, how we can do that? First, the drought sensitive high yielding cultivar will be crossed with the drought tolerant cultivar. Okay. Then in F1, one chromosome will come from parent 1 and its homologous, and another chromosome will come from the parent 2, because each chromosomes are present in 2 copies. One set will come from one parent, another set will come from another parent. In hybrid we will get both the chromosome, we can see both the chromosomes. Now, here we can see that the red gene or marker which is associated with the drought tolerance, it has been transferred into the F1. So, the F1 will be drought tolerant in nature. In next generation, as in backcross, the F1 will be crossed with the recurrent parent again and again. A recurrent parent is a drought sensitive high yielding cultivar we have used here, but it has other traits which are very favorable. It has good yield potential, good agronomic traits, other characters are fine, only it was drought sensitive. Now, once the F1 is crossed with that particular one, means once the backcross is started, then we will get different types of recombinants, means the genetic part will be exchanged due to crossing over and those things. And finally, among those recombinants, some of them will have the resistant gene, drought resistant gene like red one, and some of them will not have that part. Okay. But we have to choose those plants where maximum part is coming from the drought sensitive cultivar along with the red part, as it has been shown over here, because in rest of them along with the drought resistant trait, drought resistant part, very less amount of P1 parent's genome have been recovered. So, this recovery can be easily done through foreground and background selection. Once we will select for our targeted trait using the red colored region, using the marker specific to red colored region, this is known as the foreground selection. But if we have to recover the rest of the part of the genome of the high yielding plant's genome, that is the blue region, for that reason we have to go for background selection. So, in this way, by using foreground and background selection, we can transfer all the desirable traits from high yielding cultivar and the drought resistant trait from the low yielder, and we can get our desirable product very easily without a successive generations of yield trial. So, thereafter that particular type what we have chosen here, that plants will be crossed again, means this crossing will be done at least 3 to 4 generations, at least 3 to 4 generations back cross is needed, and thereafter finally we can get a product like this one, where most of the part have been recovered from the high yielding cultivar, but the drought tolerant gene has been integrated over there. So, in this way, by using foreground and background selection, we can get a suitable genotype where from the good plant most of the genome has come, but only the resistant gene has come from the donor parent. So, this will be the product of marker assisted backcrossing. In P1 you can see the drought sensitive high yielding cultivar, it is semi dwarf in nature, its root growth is poor because it is sensitive to drought stress. Whereas in P2, which is the drought tolerant cultivar, it is taller, its vegetative growth is huge, maybe reproductive growth is lesser, but it has profuse root growth. So, after marker assisted backcrossing, finally we have developed a plant, we can develop a plant where the root part has come from the drought tolerant cultivar, okay, and it has been semi dwarf in nature also. So, in this way we can generate a drought tolerant cultivar through marker assisted backcrossing. Okay. In case of rice, earlier flooding was a huge problem, even nowadays the flooding is a huge problem, and Sorno, which is a mega variety which is grown across the India, almost in the 80% farm, the Sorno variety is grown. There also a gene has been introgressed by backcrossing, okay, and Sorno sub 1 has been developed. Similarly, for combating the salt stress, the saltol gene have been introgressed into the rice genome, okay, and to make the plant more tolerant to salt stress, to make the rice plant more tolerant to salt stress. So, these are the importance of marker assisted plant breeding. First one is that it is highly reproducible in nature. In conventional plant breeding, what we do, we have to depend on the environment, we have to grow all the plants, all the breeding materials, we have to grow in the fields, and we have to screen very meticulously, okay. But here we can get very high reproducibility, because if you screen for disease resistance in the field, every year the climate will not be same, In sometimes it will be rainy season, in sometimes the weather may be too much dry, so the disease development will not be adequate. So, in that case the reproducibility is not too much in case of normal phenotypic analysis by conventional breeding method. But if we do the marker assisted breeding, if you know about the DNA sequence, then the reproducibility will be more. Okay. Then it is less time consuming, as you have seen that in case of conventional backcross method, at least 12 to 14 years is needed to develop a particular backcrossed line, a variety. Whereas, if you go through the marker assisted breeding, here by 6 to 8 years you can develop a particular trait. Why it is so? Because in conventional breeding you have to do backcrossing with the recurrent parent for at least 6 generations, and after 6 generations of backcrossing you are doing selfing like that. So, why you are doing 6 generations of backcrossing? So that the genome of the donor parent is recovered, maximum genome of the donor parent maximum genome of the recurrent parent is recovered, okay, along with the specific trait from the donor parent. So, here the same thing can be done using molecular gene sequences or molecular marker. So, in very short period of time, in 3 to 4 generations we can screen it, that okay, among these 1000 plants, these 4 plants have 90% genome from our desirable parent, and it has the resistant trait also. So, in this way we can choose our target very easily in less time. Next one, the early detection of traits. Suppose we have to do backcrossing for earliness in a plant or for flower color. So, in that case, once we grow the plants, we have to wait until it's come in the reproductive stage. But if you know the molecular marker, if you know the gene sequences associated with different traits, then we can do at seedling stage also. Just we have to isolate the DNA, we have to cross it, we have to do either PCR based analysis or hybridization based analysis, and in this way we can screen at very early stage. Small sample is needed. For conventional plant breeding, a huge field is needed to maintain the segregating population, because we cannot remove a promising lines. But in case of marker assisted breeding, we can screen very easily using the molecular marker in our lab, because in a small place the plants can be grown. At early stage we can screen and we can remove the unnecessary plants from there. So, very small amount of sample is needed. Next, it is cost effective. The labor cost will be minimized because we do not have to maintain the huge field. Here we can depend on the laboratory work along with the maintenance of the polyhouse or small area. Next one is the recessive alleles can be identified. In conventional plant breeding, as I mentioned earlier, the dominant allele and recessive allele, the dominant allele is those allele which can show its character, okay? Whereas the recessive allele does not show its character by itself. Let us assume in case of plant height, the capital A is the capital A allele is responsible for plant height. So, the capital A plant will be taller in nature, and the small a plant is dwarf in nature. So, if we have two seedlings and we do not know its genotype, so at early stage we cannot say that it will be taller and it will be dwarf in nature. So, at DNA level, if we know that the small a allele will be like this one, this type of molecular marker is associated with that allele. So, we can screen at very early stage, and the recessive alleles can also be identified through marker assisted breeding. So, these are the references what we have covered mostly. where from we have mostly discussed. First one is Singh and Singh, and next one is Singh in 2003, and from the web reference we have used a particular database from the plant cell, and that is all for today's discussion. Thank you.